So the first bit here is about quantifying proteins, and the main reason we can quantify proteins fairly easily is very often they absorb in visible or ultraviolet regions. So if we take this protein here and we could think back to our work on color wheels, then if my substance is absorbing around about here, which is kind of this region, then the light that's not used I'm going to see as a color, and so that substance would have a kind of bluey appearance. And in a later presentation we'll talk about where that color is coming from, and it's coming from a phenomenon known as conjugation, where we've got these alternating double and single bonds, and we're seeing that shifts the absorbance to longer wavelengths. And you can see a couple of examples there where there's a little bit of conjugation there, but much more conjugation there, and you can see we're absorbing at a longer wavelength. So if we look at something like chlorophyll, A and B, you can see in here a nice alternating pattern of single and double bonds, quite a lot of conjugation. So it's absorbing in the visible spectrum, and you can see that chlorophyll B is absorbing over here, chlorophyll A is absorbing over here, and as a result we're left with leaves appearing green. So these are trapping blue parts of visible light and orangey red parts of the visible spectrum, and using that in the plants to make energy. We're also using this idea of blocking out light, absorbing light in sun creams, where we've got some degree of conjugation here, and that will protect us from some ultraviolet radiation. And we're also making use of it when we do a titration. So if we think of phenolphthalein, it's got two forms here. There's this equilibrium existing depending on whether we're protonated or deprotonated. If we're in acid, hopefully you can see there's very little conjugation. So I've got conjugation in the benzene ring, but that's about it. But when I start to add alkali to it, if I tip the equilibrium this way, then you can now see this again alternating single and double system. And so I've got more conjugation, it's absorbing in the visible region, and as a result I'm seeing a colour being produced. So in terms of actually quantifying it, we can make use of this Beer-Lambert law, and this equation's in your data books, but essentially it's telling you that the absorbance, which is this, it's a logarithm of the ratio of the incident radiation to what's transmitted, or the intensity I should say, is proportional to the concentration of your substance. Now this value here is typically 1 because it's the path length in your cuvette, so the light's moving through a sample that's usually 1 centimeter long, and this is the molar absorptivity that's specific to that substance at a particular wavelength. But this proportionality means that we can use these calibration curves. So if I take some known concentrations of my protein and I measure the absorbance, and I end up with maybe these 5 data points at 6, I'm going through 0 through the origin, and then I take my unknown sample and it turns out the absorbance is 0.5, then I can from that work out what its concentration is going to be. Right? Buffering, amino acids can act as buffers. Talked about them being amphiprotic earlier. You can see them here gaining and losing protons. How does a buffer work more generally? Well, 2 buffer systems we can consider. One would be a weak acid and its salt, or a weak base and its salt. And if we think of our equilibrium expression for the weak acid to begin with, then we know that not very much of this dissociates. So I've got lots and lots of that substance. And also, because I've got my salt in there, that when it goes into water is completely going to dissociate into these substances. So I've also got a healthy amount of that present as well. So I've got lots of this, lots of this. So what happens if I add in some OH minus ions, Well, we can either think of them reacting with this, or we can talk about them reacting with an H+. It's easy to look at that perhaps. So they react with the H+, that's going to give us water. So my OH minus has been taken care of, and then this equilibrium is going to shift that way to replace it, and as a result my pH isn't going to change. Definition of a buffer being, of course, something that's able to withstand changes in pH when small amounts of acid or alkali are added. So what happens if I add in H+? Well, that H+ is going to combine with that to give me ethanoic acid, which, because it's a weak acid, doesn't dissociate very much at all, and as a result that has been taken care of as well. Down here with my alkali buffer, so if I wanted a buffer system, say I was doing one of those gel electrophoresis things and I wanted a buffer that was around about pH 9, I'd look to mix these chemicals together. And what I have here then is lots of ammonia, because it's a weak base, it's not going to do much by way of dissociation. But then my salt, as soon as that goes in the water, I get lots of NH4+ and chloride ions. So what happens if I put in H pluses? Well, that's going to react with whatever ones of those it can find to give me water. My equilibrium then shifts across to replace them. If I add in OH minus ions, that's going to react with this stuff, and that's going to generate back ammonia. We know that ammonia, being a weak base, isn't going to dissociate very much itself. So again, I've taken care of these ions and my system's acting as a buffer. Calculating the pH of a buffer system, you can start from Ka or indeed Kb expressions and think, well, what's in this equation? So if I, just as an example, think of that ethanoic acid, this is what the Ka expression would look like broadly. But where are these terms actually coming from? Well, this A minus on the top, that's going to be coming from our salt, right? That's what was giving us most of that A minus. And then this is my weak acid. We're assuming very little dissociation. So by knowing the Ka of the weak acid, I can solve it for H+. But in your data booklet, this equation's been expanded, the Henderson-Hasselbalch equation, and it's maybe a more friendly way of using this, because now you just have to put the numbers in. So if you've got your salt, that would be the sodium ethanoate that we're dealing with. This is your weak acid, the ethanoic acid in the last example, and you just solve it. If we were using an alkali buffer, then this is the equation I'd use, and that would give me POH. So this one, and I'm using PKB. So just this is in your data booklet. This one isn't, but hopefully you can see that the two are related. So sample calculation here, using that expression, and if we start with our Henderson-Hasselbalch equation, pKa plus log of A minus over HA like that. pKa, so P means minus log 10 of what follows, so that's 4.76, plus the log of the concentration of the salt, and because it's in 1 decimeter cubed, that number of moles is the concentration. There we go, and solving that, I get the answer, 4.91. Right, a little bit more on enzymes. We've already talked about their intricate active sites, so these folding of the protein chain, giving these substrates that are exposed, and we've got a definite fit for our substrate going in there. Example being chymotrypsin, and again we've already looked at this. You can see that we need a very specific shaped substrate to fit into this pocket, and then this is where the actual reaction occurs. Now we can study rates of enzyme reactions using something called enzyme kinetics, and if we think of our previous work on rates of reaction, you'll know that increasing concentration increases rate because we have a higher collision frequency, and we can see that for our enzymes. However, it eventually levels off for an enzyme, and we get to this maximum rate, and that's where all of our active sites are full and they're catalyzing the reaction as quickly as it can. So one thing we can discuss in terms of an enzyme is its Vmax, the fastest rate at which it can work. Related to Vmax, at half of Vmax, we can define something called, so the Km is a measure of the affinity of our enzyme for the substrate, and you can see it's measured as it's going to be units of concentration coming down here, and the higher the value of Km, the lower the affinity. So a more efficient enzyme is going to have a lower value of Km. Now enzymes can be inhibited, and there's two types of inhibition to look at. Competitive inhibition, where our inhibitor, similar shape to the substrate, and it's competing for the same active site. Can also get non-competitive inhibition, where my inhibitor is binding somewhere else on the protein, something called an allosteric site, and it's changing the shape of the active site and maybe stopping the catalysis from taking place. So competitive and non-competitive. And we can identify what type of competition we've got going on based on how it affects our kinetics plots. So this is competitive, and a telltale indicator that we've got competitive is that our Vmaxes will be the same. Right, we can get to the same point of Vmax, because if we put lots and lots and lots and lots of substrate in, we're going to manage to fill all the active sites again, and as a result we can get to the same Vmax. However, we see that the affinity is less, my Km has shifted. If it's non-competitive, then I see a drop in my Vmax, because as long as there's some inhibitor there, it's going to stop a certain amount of those enzymes from working. So some of those active sites aren't going to be accessible. However, when the substrate does go in, the affinity is just as good, and so my Km is unchanged.